Okay, folks, how's everybody doing tonight? The brain's going very slow.
No, it's going to explode.
Explode? Oh.
Like cramming in more biochemistry.
Well, could be a new energy source, you know. Have you thought about that? The more exploding brains we have, the more we could capture that energy in electricity or something, right? One small life for man, one giant explosion for mankind. So, how's it feeling? A lot of stuff? A lot of stuff. Second exam usually has a lot of stuff. Yeah. What's that? Well, you guys set the curve. I don't set the curve, so. What's that?
Still got to kick her out.
Kick her out. Okay, well, it's better than kicking me out, I guess. So, I guess what I will tell you is what I said on the last exam, and that is that I really do try to hit the high points. I try not to do obscure things. I try to make it things that are important that we've talked about and that I think are important to understand about biochemistry. There are, many things to learn, and I'm a big advocate of writing things down. So on the final exam, you will get a note card that you can bring in and fill out and use, and I think you'll find when you do that you will actually not use it much because the filling out of the note card actually helps you a lot to organize that in your heads. So if I were to advise you on this, I would say, well, practice by filling out a note card for this one. And I think you'll find it actually does help you to organize things very well. There is a lot of information. I'll be very honest with that. There's a lot of information on this exam. The good news is, as you know, I don't make you memorize very many structures, just a handful of structures to know. And that'll be true next term as well. I hope that, and of course today's material won't be on the exam, but I hope that you saw as a result of today's material that metabolism is something that's pretty interesting, and you can learn a lot about how organisms work by understanding metabolism. And as I've alluded to before, that will only grow as we talk more and more about metabolism. So we finished this year talking about sugar metabolism, which is glycolysis now, gluconeogenesis, and then we'll be talking about glycogen metabolism. And so some pretty exciting stuff to come ahead. So a lot more things to learn. All right, so I said we start at 5:15, so it's 5:15, and I'm ready to start if you guys are. Yeah?
I came from recitation when
Okay.
TA did a really nice review.
Okay.
And she brought one thing up where I was like, what? And it was the myosins, the ATP-dependent motor protein.
Yeah.
Is that?
Yeah.
Do you think that's something you'll?
Do I think myosins is something I'll ask you about? Well, I can't tell you what I'm going to ask about. If I did, I'd have a very popular video. My guideline is always, look at what I talked about in class. What I talk about in class is really the bottom line of what's there. I didn't spend much time talking about myosins, so if I were to ask you about myosins, there wouldn't be a lot of things I could ask you, but I did talk about them. So, does that make sense? Yeah. Kind of a cop-out answer, isn't it? Yes, sir.
Can you, remind us where the cutoff was of the material for exam 1? I'm trying to remember where it kind of started.
Well, it's actually marked on the highlights. If you look on the highlights, you'll see exactly where it goes. Yeah. Yep.
I think it was step 7.
Hi. Yeah.
Can you go over things called dolichol phosphate again?
Can I go over dolichol phosphate again? Sure. Let's take a look at dolichol phosphate. So I'll bring up the figure just to show you what it looks like. And if I can remember where it was, I think it was in carbohydrates, right? Yep. And when we talk about dolichol phosphate, right here. Okay, so dolichol phosphate is an odd-looking molecule. It has a long nonpolar tail, and it has a phosphate sticking off the end. That's what's sticking off the end over here off of the screen. Okay? And it turns out to be an important molecule for decorating, that's the term I gave, for putting on those oligosaccharides onto glycoproteins. It's an important molecule for adding those oligosaccharides onto glycoproteins. All right. So specifically, this molecule is found in the membrane of the endoplasmic reticulum. And you recall that in the endoplasmic reticulum, the N-linked glycoproteins, that's where they start getting made. So this is a molecule that helps the N-linked glycoproteins to get their glyco part. That is the oligosaccharide that goes onto them. And the way this works is this guy sits in the membrane of the endoplasmic reticulum, pointing out into the cytoplasm. So out of the endoplasmic reticulum means into the cytoplasm. On the surface, at that place where that phosphate is, okay, at the place where the phosphate is, that's where this sort of core of oligosaccharides gets built. And I'll show you that core again, just so you know that. All right. So the core of oligosaccharides looks like this guy right here, this Y. So these first 5, 1, 2, 3, 4, 5, those are common to the N-linked glycoproteins, all right? And that core is built on the Dolichol phosphate, okay? So Dolichol phosphate has that. Then, after that core has been put onto there, as I said in class, this Dolichol phosphate flips, and when it flips, it brings that core, which is attached to it, to the inside of the endoplasmic reticulum. At that point, it gets transferred onto the glycoprotein, onto the asparagine residues, and then the glycoprotein gets other things put onto it after that point. So the Dolichol phosphate's role is to provide a sort of attachment point for that oligosaccharide. The building starts on the outside, moves to the inside, and then gets transferred to the glycoprotein. Yes?
So you're saying it, I thought the glyco part of the protein was when the oligosaccharide was put on it.
Say it again?
So the glyco part of the glycoprotein is the oligosaccharide.
The glyco part of the glycoprotein is the oligosaccharide. That's
Okay.
correct.
So then it actually becomes the glycoprotein once Dolichol phosphate turns it into the ER.
So once it gets taken off of the Dolichol phosphate and put onto the protein, it becomes a glycoprotein. Make sense? All right. I wasn't sure if this room would be big enough, but fortunately it looks like it is big enough. So unless we get a giant influx of people in the next 5 minutes. Okay, other questions? Yes?
Can viral DNA that has been inserted into bacterial DNA become methylated as well, or is it only bacterial?
Oh, that's a good question. So I talked about restriction endonuclease, restriction modification systems, and I talked about the methylases that can put methyl groups onto the same recognition sequences that the restriction enzymes themselves cut. And her question has to do with and the reason that that's done is to protect the cellular DNA. So the viral DNA that comes in is not methylated, and if the restriction enzyme encounters the viral DNA, it will cut up the viral DNA, excuse me, but not the cellular DNA. And her question is, can the viral DNA itself get methylated? And the answer is yes, it can. So as I said in class, it's not a perfect system. It's a system that works most of the time, but even our own immune system is not perfect. So this protection that the restriction modification system gives to the bacterium is definitely advantageous, but it doesn't stop every single virus, just as we don't stop every single virus. Okay, so. Yes?
Can you actually talk about EcoR5, just preventing the breakdown?
Okay, so let's talk. A question has to do with, can I talk about EcoR5? So let's go to EcoR5 and talk about that. So going back to catalytic strategies and talking about EcoR5. Let's see, where do I want? I'm the wrong thing, aren't I? Right here. Okay. So, EcoR5, excuse me, recognizes the sequence GAT-ATC. You don't need to know that. But like other restriction enzymes, what it does is it finds its target sequence by getting onto the DNA, and it literally just grabs a hold of the DNA, and then it starts sliding down the DNA. And as it slides down the DNA, if it encounters its recognition site, it stops. If it doesn't encounter its recognition site, it just keeps sliding down the DNA. So it finds its recognition site and stops, okay? Then, like we talked about with the serine proteases, binding of the appropriate substrate, which in this case is the recognition site, binding of the appropriate substrate causes the enzyme itself to undergo a physical change in shape. And that physical change in shape, that physical change in shape that it undergoes helps to bend the DNA right in the middle of that recognition site. Okay? That bend, okay, will not happen, and the recognition site will not be recognized if there's a methyl group there. So if that sequence had been methylated, then that enzyme just goes sliding along and doesn't do anything to that recognition site, which is why methylation protects the recognition site from being cut in cellular DNA. Okay, so if there's no methyl group there and it's found its site, the enzyme has undergone a change in shape. That change in shape bends DNA, and that bend in DNA actually facilitates the orientation of a magnesium ion that helps to hold a water ion in place. And so we're thinking about geometry here. Geometry is very critical. This bend is necessary for this little pocket to be formed in order for this overall structure to be created. That water loses a hydrogen, and when it loses a proton, it becomes a hydroxyl, it becomes a nucleophile, and it attacks the phosphodiester bond. That attack of the phosphodiester bond is necessary and sufficient for the cutting of the DNA, so long as both strands gets cut. And yes, both strands will get cut in this process. So that's what's happening with the EcoR5. And this is true of other restriction enzymes as well.
The group that's on there, is that basically a metalloprotease?
The group that's on there on the...
Because remember, we were talking about metalloprotease, it's sort of the same thing. You have like a metal that's holding water in place, and the water facilitates the breaking of the carbonyl.
Yeah. So restriction endonucleases are not metalloproteases because obviously for one, they're not proteases because they're cutting DNA. But they act like metalloproteases in the sense that they are, in fact, creating nucleophiles that are reactive and that attack in this case, the phosphodiester bond.
Very similar math.
Well, in a nucleophilic attack. So all nucleophilic attacks are going to have that sort of same similarity, yes. Yes? Does the magnesium pull the proton off of water? No, it doesn't. Magnesium, kind of like you're talking about in metalloproteases, is helping to orient this overall water in the right place so that the water can ionize and then make the attack. Yes?
So you're having bending of the EcoR5 and DNA, or just the DNA?
Well, the EcoR5 is holding the DNA, so since it's holding the DNA is being bent as a result.
Does the EcoR5 undergo conformational change first with causes the DNA?
Well, imagine that I've got the DNA in my grasp, and all of a sudden my hand changes. The structure of the DNA is going to change simultaneous with it, right? Yeah. Yeah.
You said that the methylation affects the orientation of the water and the metal
Yes.
inside.
Mm-hmm.
And I was wondering, if that's the case, why would it slide right across if the methylation was there on the recognition side?
Okay. So this question has to do with why is it that methylation stops the recognition? Okay. And this is a little bit more advanced than we are, but I'll tell you briefly what the answer to that question is. Okay. So the methyl group provides a steric hindrance. It's actually something that's there's a hole there before, and there's not a hole there when you put a methyl group in there. And in order for a DNA recognition enzyme to recognize its sequence, what it's actually recognizing is a pattern of hydrogen bonds. So it's looking for that pattern, the right pattern of hydrogen bonds that tells us, oh, here's a GAT-ATC. Anything that interferes with that process is going to interfere with it recognizing something as GAT-ATC. So in simple terms, what the methyl group is doing is it's providing a steric hindrance that doesn't allow the proper reading of hydrogen bonds. Make sense? Okay. Yeah.
So for the water to ionize, does that occur in a higher pH level, similar to the?
Yeah, good question. For the water to ionize, does it mean it has to occur at a higher pH level? That's a good question. I know of some enzymes that work really well at pH 9. So you would argue that that's the case. I can't speak that this mechanism is exactly the same for all enzymes. There are hundreds of restriction endonucleases, and I can imagine that some of them don't involve the same mechanism. Okay, but EcoR5 definitely uses this.
I guess we should just know that it ionizes.
Yeah. What? Yeah. That's right. So I mean, it's a good question. And when we think about enzymes, okay, we have really two things to think about. One is the one that you're thinking about, which is we've got an intermediate that we would really like to, make more favorable to form, which would happen at a higher pH. But we also remember that enzymes are sensitive to pH, and so while the intermediate might very well form, let's say the intermediate will form great at pH 12, but the enzyme, won't tolerate that. There's a balance that happens in any enzyme in terms of optimal conditions and the conditions that it's stable at. So the good example was carbonic anhydrase. And you saw that it went through to 9. And like I said in class, if you went above 9, you would start seeing the enzyme doing this because it's denaturing. And so the same thing would happen here. Okay? Yes, Eric? And the what? Her? Okay, yeah. So the question about EGF and her. So her is a receptor. An EGF receptor is a receptor. All right? And so, as I said in class, these two receptors can interact with each other, form what would be the equivalent of an EGF receptor dimer. Okay? And when that dimer forms, then you see activation of the processes that lead ultimately to cell division. So, as I said in class, at a low level, this is probably happening all the time, not a problem. The time when her is a problem is when something happens that changes its expression and the cell starts making a lot of her. Cell starts making a lot of her, it's really hard for the cell to properly control that, and uncontrolled growth arises. But these are the receptors we're talking about, not the EGF hormone. Okay? Yes?
Can we go over the signaling process with the EGF?
Can we go over the signaling process with the EGF? Sure. So if we go back to here and to here. Nope. I think it's here. Yep. Okay. So this depicts the signaling process that's going through the EGF receptor up to the point of activation of RAS. As I said in class, this activated RAS now will go and interact with a variety of things and can help to stimulate the division of cells. The activation of this pathway starts with the binding of EGF receptor on the surface. Binding of EGF on the surface of the EGF receptor. And as you can see, in order to put two EGF receptors together, which is necessary for the activation of this pathway, in order for that to happen, each of the EGF receptors has to bind an EGF. You put two EGF receptors together like this, and these carboxy tails that are down here are full of tyrosines. And moreover, each of these inner components is a tyrosine kinase. The individual receptors can't phosphorylate themselves, but when brought into close proximity of another one, they can then phosphorylate the other one. Well, the phosphorylation actually has a couple of effects, one of which is, as you see here, the recognition of those phosphotyrosines by other complexes. But the other, and I didn't say this in class, but I'll say it here just so that you know, is that the phosphorylation of these tyrosines actually helps to make this thing even more active. So it's kind of like a jump start for everything. This receptor becomes more active as a tyrosine protein kinase. For our purposes, that didn't really matter because right here we see that we've got the phosphorylated tyrosine, which is a binding site for the SH2 domain of these other proteins that are involved in the overall signaling. Okay? So we see, for example, the binding here of the GERB2, and the GERB2 is itself recognized by this protein SOS, which in turn is recognized by this protein RAS. And so the sort of binding that's happening with these proteins is happening as a sort of a chain reaction you can think about, and it's stimulated ultimately by this. This causes this to bind. Binding of this now facilitates the binding of this. Binding of this facilitates the dropping of GDP out of RAS and the loading of GTP in its place. Okay? Yes?
So in all of these signaling processes, when we have phosphorylated tyrosine, are they always bonded to those domains?
So his question is, are SH2 domains the only things that bind to phosphotyrosines? There are other things that will bind to phosphotyrosines. Okay, but we talked about SH2 domains here. Okay. Anyway, the RAS will drop its GDP, pick up GTP. That has the effect of activating RAS. Activated RAS then can go through the cell and stimulate, ultimately, the division of the cell. RAS, you remember, is as I described, a poor enzyme. It is able to break GTP down to GDP, but it takes on the order of minutes for that to happen. And that is good because it allows for RAS to communicate its signal during that time. The bad news is if the GTP breakdown part of RAS is inactivated, RAS can still act because it now has GTP bound to it. It thinks it needs to turn everything on, but it can't break the GTP down. And that, in turn, causes RAS to activate cell division when it shouldn't be activating cell division, and that results or can result in a tumor, uncontrolled growth. Yes?
Are these GERB2, SOS, and RAS, are they membrane bound or are they free floating in the cytoplasm?
Yeah, that's a good question. They're pretty much membrane bound or membrane associated.
Except for when RAS gets a GTP, in which case it can then dissociate?
RAS actually is pretty much membrane bound as well. So RAS actually has on it a fatty acid that anchors it to the membrane.
Okay, so I guess this is where I was getting confused. I thought that GTP-bound RAS went throughout the cell, then activated the other proteins.
No. So its effect goes throughout the cell, but RAS physically doesn't go throughout the cell. RAS is pretty much membrane bound, and RAS can activate, let's say you saw how, for example, protein kinase C. Protein kinase C got activated. Protein kinase C can go do its thing around the cell. So all it takes is RAS activating one protein that can go float in the cell so that the effect goes into the cell. But RAS is pretty much membrane bound. Yes? Is RAS a second messenger? No, because it's not synthesized in response to this. RAS is already there. So a second messenger is something that's synthesized in response to a signal. So we don't see anything here that's been synthesized. Okay? So we're not talking about a second messenger in here. Yes, sir?
We talked a little about 7TM receptors.
7TM receptors, sure. So go back and remind you a little bit about 7TM receptors. So 7TM receptors are a class, and I find students, it's interesting, I talk to a lot of students, and I find students have a real difficult time sorting out an individual of a group and a group. Okay? So, for example, I was talking to somebody the other day, and they had confusion with what a protease was. A protease a specific thing, or was it a group? And was a chymotrypsin a specific thing or a group? And maybe I'm not being clear about these, so maybe I should spend more time on them. But it's important in organizing all this information in your heads and so forth to be able to recognize categories of things. Okay. Well, I mentioned that here because 7TMs it's a category. It's a category of membrane proteins that have a common structure and that many of which have common functions in signaling. Okay? So there are hundreds of these. All right? There are many, many 7-TMs, and they're found in membranes. They do have commonality of structure, as I noted, and the single commonality that they all have is that they have 7 transmembrane domains. They have parts of them that go from the outside to the inside, to the inside, to the outside, to the outside, to the inside. And so this is the polypeptide chain that is that protein. And so it's going in this up, down, around, 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 around route 7 times in the membrane. Okay? That means, therefore, that the amino acids that are exposed here are going to be mostly hydrophobic because this part of the membrane is very hydrophobic. Okay? So it's actually relatively easy for us to take a DNA sequence and analyze the sequence and look for regions of nonpolar amino acids and ask the question, could this be a 7-TM? Do we see 7 of these? And if you look, the 7 is separated by these little things here and the little thing here. Well, what's the nature of those little things? The nature of those little things is they're going to be very hydrophilic because it's interacting with the cytoplasm down here, which is water, or it's interacting with the outside of the cell, which is also water. So identifying these is pretty straightforward. All right, you can say, does it have about the right number to cross the membrane? Are they hydrophilic? Is it followed by a short? Are they hydrophobic? Is it followed by a short hydrophilic? And then the same thing and the same thing. So a computer can actually tell you with reasonable accuracy how whether a given protein is a 7-TM or not, more than you wanted to know. Now, 7-TM, there are many 7-TMs that are involved in signaling processes in the cell. We talked about 2 of them. Indira talked about both of them. I finished up one of them. And the first of those was the beta-adrenergic receptor. So the beta-adrenergic receptor was a 7-TM. And if we look at the 7-TM from a different perspective, okay, this is a more accurate way that it actually appears inside the membrane. It's not all lined up and 2-dimensional like you saw in the last slide, but it actually looks more like this. This 7-TM, in the case of the beta-adrenergic receptor, binds to the hormone known as epinephrine. And all of these 7-TMs have a common feature that when they bind, they, in fact, change shape, which is really good. That change of shape, as you've seen, is what's critical for communicating information or making something happen. In this case, it's communicating information because the inside part is changed in a way that its interaction with the G protein is altered. That interaction with the G protein, in the case of the beta-adrenergic receptor, causes the G protein to lose GDP, replace it with GTP, and then that G protein goes and interacts with the adenylate cyclase. And if you look at the pathway for that, where's it at? Here it is. Okay? So here's what's happened so far. The binding of the hormone up here, the unloading of the GDP, replacement of GTP, that causes the beta and the gamma units to go flying away. That exposes on here a binding site for the adenylate cyclase. The adenylate cyclase, when it binds to this guy here, makes ATP. And ATP, I'm sorry, it doesn't make ATP, it makes cyclic AMP. It converts the ATP into cyclic AMP. And then cyclic AMP, in turn, causes protein kinase A to become activated. This goes and phosphorylates a variety of proteins. It's important for this pathway to be turned off. And Indira talked a little bit about the ways this pathway can be turned off. But one of the ways is, again, the fact that this guy can cleave GTP, just like RAS can. So it's a bad GTPase, but it cuts the bad meaning inefficient, and it cuts GTP, makes GDP, turns itself off. Okay, so that stops part of the pathway when that happens. All right. The other parts of the pathway is we want to turn this business off as well, because if we have this guy turn itself off, but it comes back over here and gets reactivated, it's not going to accomplish anything. So when we think about how the rest of that pathway gets turned off, okay, there's a couple ways in which that can happen. All right. So one is actually that's how So one is that the GTP, as I said here, is breaking off the phosphate to make GDP that causes it to leave this thing, pick up its beta gamma units, and it's back where it started. That is part of turning off the process. The other part of turning off the process is shown over here. So here was that activated the activated beta-adrenergic receptor because it's got the epinephrine in it. It can be turned off in two different ways. One is it can simply let go of and lose the epinephrine. And remember, all these bindings with proteins and molecules are reversible. These are all reversible processes. So it can let go of this. This goes flying away somewhere. And we end up in this state where it's turned off. It's not going to activate any more G proteins. If for some reason it can't get rid of this, okay, like let's say there was some problem, it got stuck or whatever, we still want to have a way of turning this off, because if we don't have a way of turning this off, what's going to happen is the cell's going to burn up all of its glucose reserves, or all of its energy reserves, I should say. And this pathway going this direction is a backup way to turn off this thing. Okay? When this thing is stuck in this state, there's a receptor kinase that phosphorylates the end of the beta-adrenergic receptor, as you can see here. That forms a target for a protein called beta-arrestin that basically gloms onto there and shuts the whole thing down. So either this or this will have the effect of turning off the beta-adrenergic receptor and, of course, stopping the 7-TM from doing its thing. Yes, ma'am?
I guess if you're releasing the epinephrine, couldn't it then go bind to other receptors? And
Could.
continue to signal?
So can the epinephrine go bind to something else? It could, yeah.
So would that, I guess, would that amplify the signal if it was able to bind to something before these other alpha subunit hydrolyzes?
So it's a good question. The answer is yes, it could, but we want to keep in mind that epinephrine itself can be broken down as well. We haven't talked about that, but yes, if what you said was right, that is, it stimulates this cell, and while that cell is still getting its stimulation, the epinephrine goes and binds to another cell and causes that to happen. It certainly could amplify the signal. Yes. Yes? Yeah, good question. Is the beta-arrestin specific to the beta-adrenergic receptor? You know, I don't know the answer to that question. I suspect it's not specific to it. Yep. I don't think it's specific, but I'm not sure of that.
So once the 7-TM is with receptors in this state with the arrestin, beta-arrestin, how does it become active again? Or is it
Once it's in this state?
Yeah.
Well, that's a good question. Again, when cells have a way of turning something on, they want to have a way of turning something off. I look at this as kind of a dead end because it's got these phosphates on here. Could those be removed by a phosphatase? Yeah, presumably they could. Another thing to keep in mind is that cells don't have just one beta-adrenergic receptor in their membrane. They may have thousands. So if you had one or two, or you had a few of these that were turned off and they couldn't be turned back on, probably wouldn't be a big deal.
And also the receptor kinase, will that act on any of the receptors that have the epinephrine bound to it for a certain amount of time?
So it will act on anything that's got epinephrine bound to it for a period of time. That's correct.
So what would control having the epinephrine bound to it for long enough? Like what if it wasn't bound long enough to cause the signal to be active?
So if epinephrine wasn't bound long enough, then we're back over here at this state, and it's not needed. Right? Yes?
Indira mentioned the fourth method, phosphodiesterase, to break down CAMP.
Yeah. So Indira's lecture, she described it as a fourth method, but it's actually another step in the overall process. So when we think about this, we want to make sure we shut off the entire signal. So I describe it differently than she does in that respect. She called it a fourth method. But what I'm saying is if we want to turn off the signal, we've got to do several things. One is we've got to turn off the receptor. If we don't turn off the receptor, we're still going to stimulate things going on, and nothing else is going to matter. So this has got to be turned off. We turn this guy off. We've also got to turn off the G protein, and the G protein does a very good job of turning itself off. So that's the second step in the turn-off process. This is the first step. The second step is turning off the G protein. The third step is we've got to get rid of that cyclic AMP that's in the cell. Because if we turn everything off and we still have a ton of cyclic AMP in the cell, we'll still always have protein kinase on, right? So the third step that has to happen is we've got to break down that cyclic AMP, and cells, kind of like they have a way of turning off the G protein, have a way of continually breaking down cyclic AMP, and that's the phosphodiesterase that's there. So cells always have a phosphodiesterase in them. And when we see a signal coming along like we see here, what happens is the cyclic AMP, when the G protein interacts with the adenylate cyclase, cyclic AMP starts being made, cellular concentrations start to rise, okay? We start turning off these processes, and then we see cellular concentrations start to fall because phosphodiesterase takes over. That tells us that the adenylate cyclase is more able to synthesize, that is, it can make this cyclic AMP faster than phosphodiesterase can break it down. Right? Yeah?
Do cells have a constant supply of phosphodiesterase?
Yes.
Or do they don't like stimulate their...
Do cells have a constant supply of phosphodiesterase? The answer is yes, they do. Absolutely. And cyclic AMP is a molecule that is a very important molecule in signaling. So controlling that is really critical. The cell wouldn't want to wait to make phosphodiesterase to bring concentrations down, because by the time it did that, it probably would be dead.
Well, I guess what I'm asking is, does the cell control phosphodiesterase activity?
Oh, I'm sorry. I thought you meant does the cell synthesize it there. The only things that I'm aware of that affect phosphodiesterase activity is caffeine. There probably are other things that do that, but the only one I'm aware of is caffeine. So having some phosphodiesterase present in the cell that's active is probably important to start with.
So we really don't care how much phosphodiesterase there is. It has a constant supply. What we really care about is the rising supply of CAMP.
That's right. The pulse of cyclic AMP. Absolutely. Yep. Yes? Is the caffeine, yeah, that's a very good question. Is the caffeine inhibition of phosphodiesterase permanent? It's not, obviously. If it were, we'd have a cup of coffee and it would last a long time. It's a question that I wonder about myself. I don't work in this area, so I can't tell you the answer to how this thing is done. But I'm intrigued by the metabolism of caffeine because I think the answer to that question is really how it is that this is controlled. And I don't know hardly anything about, if you go looking, and I've gone looking, you don't find a lot about the metabolism of caffeine. We see the effects of caffeine, but not a lot about the metabolism of caffeine. So I don't know how that's done. But it's a very good question, one I've wondered about as well. Yeah?
I have a question on a different related subject. I want to make sure everybody's done talking about this before we move on. Did
Anybody
you have
else?
a question about this?
No.
Okay. Could you talk about alpha-1 antitrypsin and elastase role in emphysema?
Yeah, alpha-1 antitrypsin and elastase's role in emphysema. So it's a very good question also. So alpha-1 antitrypsin is an inhibitor of trypsin. Trypsin is a protease. And though I didn't talk about it in class, I'll mention it here again. We think about those zymogens, right? So we made the proteases like trypsin and chymotrypsin and elastase and so forth, made in an inactive form so that they're not harming the tissue that's making them, basically. The zymogen making the enzyme in inactive form is one way of controlling it, but yet another way of controlling it is having specific inhibitors. And so an additional protection that you have against the attack of tissue by trypsin or whatever protein are the presence of certain specific inhibitors. Alpha antitrypsin is one of those. And when it was discovered, as I said, it was named alpha-1 antitrypsin because it was found to be able to inhibit trypsin, which is what gave it its name. It was subsequently realized that the alpha-1 antitrypsin actually worked better on elastase than it did on trypsin. So the name stuck, however, so it's still called alpha-1 antitrypsin. Alpha-1 antitrypsin is found in lungs, among other places, but in the lungs, it appears to help balance the amount of elastase that is active. And I mentioned in class that having some elastase active in your lungs is good, and it's important because it does provide us with some protection against things that we're breathing. All right. whether they're bacteria, viruses, or whatever. So having a protease to chew on those is good. The problem that arises with elastase is a little bit of something is good, a lot of something is bad. And the bad is the fact that when elastase is overly active, it attacks the lung tissue, just like we had the proteases attacking the pancreas. And when it attacks the lung tissue, it causes emphysema. So imbalances in the alpha-1 antitrypsin in the lungs can contribute to the formation of emphysema. Well, what happens to alpha-1 antitrypsin is the fact that it gets oxidized. And it gets oxidized, let's see here. Where's it at? Bases. That's one pocket. Somebody see it? No, it's not it. That's not it. Well, I don't see it with my finger, but in any event, alpha-1 antitrypsin gets oxidized when people smoke. All right, and what happens when people smoke is they create what are called reactive oxygen species. Sorry, and reactive oxygen species will oxidize things indiscriminately. And so one of the things that gets oxidized is a specific methionine residue in alpha-1 antitrypsin. The oxidation of that methionine is critical because that methionine is important for the binding between the alpha-1 antitrypsin and the elastase. So if that methionine gets oxidized, which is what happens when it encounters a reactive oxygen species, then it won't bind to elastase. So the more smoking, the more reactive oxygen species, the more damaged alpha-1 antitrypsin, the more active elastase, the more likely emphysema. And that's the sort of sequence of events that's there. Yes? Sure, angiotensin receptor. So that's another 7-TM. And that one is the one that involves protein kinase C. And go back here. Okay. And right here. Okay. So the angiotensin receptor is a 7-TM. It also uses a G protein. It's not shown on here, but it also uses a G protein. So angiotensin binds to its receptor, stimulates the activation of a G protein. This activation of a G protein activates an enzyme known as phospholipase C. Okay? Phospholipase C, again, the name tells you something about what it does. Phospho, involving a phosphate. Lipase, involving some sort of fat or fatty acid. In this case, it's involving a fatty acid. And the C, of course, is like ABC. So the cleavage of a molecule called PIP2, that's a phospholipid. The cleavage of PIP2 by phospholipase C results in the production of two second messengers. First of all, this small piece right here is released, and it is completely water-soluble. It's called IP3, and it's got the 3 stands for 3 phosphates. So it's very, very charged, and it's soluble in the cytoplasm. It leaves the membrane where it was and travels into the cytoplasm where it encounters the endoplasmic reticulum. The endoplasmic reticulum has a receptor that binds to IP3 very nicely, and the binding of IP3 by the receptor causes the receptor to open. It's like a little channel. We'll talk about channels next term. But the little channel opens, and when it opens, it provides a way for calcium ions to escape from the inside of the endoplasmic reticulum. I mentioned in class that the calcium is problematic in the cell, which is why it is sequestered, as you see it sequestered here. Leaking of a little bit out doesn't cause a problem, but it does start a signal, or it continues a signal process occurring. So we've got a second messenger that's come down here. calcium is what I called a third messenger, although you can call it either second or third as far as I'm concerned. And the other second messenger is up here. What was left behind when we cut off that IP3 is something called DAG, D-A-G, diacylglycerol. That DAG remains in the membrane because it's got those long nonpolar tails that are still associated with the membrane. And it can then be a binding site for this protein kinase C. Protein kinase C can bind to DAG. Protein kinase C can bind to calcium, and when it does, it becomes active and starts phosphorylating things. Yes?
I was wondering if you can talk a little bit about the oligosaccharides.
Well, let me make sure I answer his question up here first. Is that?
Yeah, I just.
Okay. Sure. Okay, I'm sorry. Go ahead.
I have a hard time understanding the oligosaccharide and glycoprotein and glycosaminoglycan.
Okay, so question has to do with oligosaccharides, glycoproteins, and glycosaminoglycans. So how do you distinguish those? Is that what the question is?
Yeah.
Okay. So actually, before I do that, is there any other questions on this? Yeah, okay. Sorry. Go ahead first here.
So the IP3, how does it, it's stuck on the receptor, like how does it leave the receptor so the calcium ions don't continue to
Okay. So how does it get out of there? All right. So there are things that'll break down IP3. We haven't talked about them, but it's a good question. IP3 has to be broken down. Otherwise, it'll continue to do that, and then all the calcium would leak out. We'd have a problem. Yep.
We talked about protein kinase C. We also talked about calmodulin. Calmodulin is activated by calcium as well. They're both activated by this process.
Yep.
And they go on, and there's a bunch of other things that are activated, and the net result is smooth muscle contraction.
Yes.
Did we not talk about that?
Yeah.
Specifics?
I have not.
Other than the EF hand?
Other than the EF hands, I haven't talked. I certainly haven't talked about smooth muscle contraction, other than the fact that it happens. It's one of the things we don't talk about here, and it would be the issue is that of time. I would like to spend some time talking about that. The issue of smooth, the mechanism of smooth muscle contraction is interesting.
But it is the purpose of this receptor.
It is. Yes. Yep. Yep. Right. So calmodulin, as I noted, is a way of sequestering calcium and still having the effect of the calcium ion itself signaling, but in this case, the calmodulin is kind of carrying it and keeping the calcium from being a problem. Yes. Yes. Okay. Other questions about this?
Calmodulin also have another role. Was that-
Oh, calmodulin has many roles. Yeah. So in fact, we'll see a role for calmodulin next week. Specific role.
Activate protein kinases?
There are some kinases it can activate, yes, and we'll talk about those next week. Yep. Okay, now back to your question. Okay, sorry. So your question had to do with oligosaccharides, glycoproteins, and glycosaminoglycans. So that's in the carbohydrates. Let's go back and talk about that. So I'll be honest, I probably wasn't as clear on that as I could have been. So let's try to clear that up. Let's start by talking about glycosaminoglycans. Okay? Glycosaminoglycans. What is a glycosaminoglycan? All right? Glycosaminoglycans are long polymers that are polyanionic that have modified carbohydrate residues as their repeating units. So So glycosaminoglycans are modified carbohydrate residues in a polymer, polyanionic, okay? And their polyanionic properties cause them to have some useful things that they do, okay? If we look at these, we see here's an amine. Here's a modification that allows this guy to be very negatively charged. Here's an amine. Here's some negative charge. There's an amine right there you can barely see. Negative charge. There's one over here, but you don't see it. One here, and so forth. So that's the amine part. Glycose, amino, glycan. So glyco meaning carbohydrate. Amine meaning having an amine like here. And a glycan is simply a polymer of sugar residues. Okay? So these guys have the interesting property, because they're so anionic and they repel each other so well, they actually change the properties of the water that they're dissolved in. These guys might have molecular weights on the order of several million. Okay? Really long polymers, really long stretched out things. All right? The common feature that they tend to have is they make water what I describe as slimier. Okay? These glycosaminoglycans might be attached to a protein. Okay? They don't have to be, but they might be attached to a protein. And that was what I was unclear about in class. Okay? So if they're attached to a protein, they might, if they're attached to a protein, they're actually called a proteoglycan. Okay? And this was the figure I showed. So here's these long glycosaminoglycans all repelling each other, all trying to get as far away from the others as they can, but they're all sort of held in place by this central protein unit. Okay? These guys are used for lubrication. As I said, snot is full of glycosaminoglycans, proteoglycans. Your synovial fluid, which contains hyaluronic acid, okay, is a lubricant and has some really interesting solution properties as a result of containing hyaluronic acid. For one thing, hyaluronic acid, in addition to being sort of oily and slimy, like I say, but it provides that sort of motor oil effect, as it were, if you want to think about that. In addition to that, the synovial fluid is very able to absorb shock. So it absorbs shock really well. And so if you think about what your knees are doing, every time you take a step, okay, you're providing shock to your knees, to your ankles, with every step. And if something doesn't absorb that shock, ultimately you're going to destroy that knee or that ankle. Well, people do eventually sometimes hurt their knees or their ankles, and they need a replacement or something like that. But the important thing is that without that synovial fluid, they would lose that ability much, much, much earlier. Yes, sir?
My uncle, he had bad knees, and he had a shot in his knees, and
Yep.
he said it helped. Great.
Yep.
Is that what that is?
Yeah. So when people get shots in their knees, typically what they're doing is they're getting injections of synovial fluid from somebody else. Okay? And the synovial fluid, when people are needing injections like that, they're low in synovial fluid, which is why they feel joint pain and so forth. That treatment is, number one, very expensive. It's $700 a shot or something like that. And it doesn't last forever, so you have to go back periodically and get that additional shot. If the problem is severe enough, it may require knee replacement because it's just difficult to keep enough synovial fluid in there.
So you said it has to be harvested?
It has to be harvested.
So it can't be synthesized?
There are people working on synthesizing artificial synovial fluid, but the problem is we don't know enough about the components of existing synovial fluid to have a perfect replacement at this point. Yeah. There's somebody on campus who works on it. Dr. Skip Rochford is one who's worked with synovial fluid. There's another one on campus as well. Okay. Now, so that was the first part of your question. All right, so there's the glycosaminoglycans. How does a glycosaminoglycan differ then from a glycoprotein? All right. A glycosaminoglycan is that long polymer. A glycoprotein is a protein, and it's mostly a protein. Okay? So this glycosaminoglycan might be 1 million in molecular weight. It would dwarf a protein in terms of size, but a glycoprotein is mostly protein with oligosaccharides on it. So oligo means a few. So oligosaccharides, when you put oligosaccharides onto a protein, you create a glycoprotein. So that's the difference, really. A glycosaminoglycan is a glycosaminoglycan is a polymer. A proteoglycan is a glycosaminoglycan attached to a protein. It's mostly glycosaminoglycan. Okay? A glycoprotein is mostly protein, and it's attached to a few oligosaccharides. Does that help? Okay. When we talk about glycoproteins, we talk about two kinds. We talk about those that are N-linked, and we talk about those that are O-linked. The N-linked the glycoproteins are linked through an asparagine residue, and they have a common 5-sugar core that you see right there. That's the thing that the Dolichol phosphate was involved in putting on. Glycoproteins are made, end-linked glycoproteins are made starting in the endoplasmic reticulum and finished in the Golgi apparatus. The other type of glycoproteins that we have are the O-linked glycoproteins. And the O-linked glycoproteins look like this. They're linked through the side chain of a serine or threonine up here. This would be a serine or threonine, and they're linked to, in this case, a galactose, N-acetylgalactose. All right. That linkage of the O linkage, there's the O that causes that's called the O linkage. There's the N that calls this an N linkage. This O linkage that's here is only made in the Golgi apparatus. It's not made in the endoplasmic reticulum. Yes, sir. That's a good question. Are they linked only to complex sugars and not simple sugars? I wouldn't say that you wouldn't absolutely find some simple sugars in there, but most of the sugars you find in glycoproteins are actually modified sugars. So you're correct most of the time. Yep. And some of these modifications of the sugars are really elaborate and really bizarre. There's some really weird things hanging off those proteins, and to be honest with you, I don't know why. There's a lot of things I don't know either. So. Yes?
You gave us a thought, I think when you were talking about qualia, you said like if you have just invented a phosphate inhibitor of an enzyme that wipes out the enzyme, what was going on?
Yeah.
So maybe like if that inhibition is something activated?
So if you had something that favored only the T state. Oh, okay. I thought I was repeating what you said, so sorry. It's just food for thought. Food for thought's good. What's that?
I was thinking about it too.
Food, you see, folks, you're here to think. You're here to think. All right? So that I think that we make a mistake in making too many things that are just regurgitate. All right? I think it's important for us to think no matter what it is that we're doing. And I would love to get to a point. I would love to take a class like this and give you an exam that every single question made you think. Every single question made you do that, because that's what you're going to have to do when you leave here. Right? So I think it's really good and it's important to think. Yes?
This is about
Okay.
These are the same things that can get phosphorylated to direct where proteins end up in cells, is that correct?
So can glycoproteins get phosphorylated? Is that the question?
Well, I'm just trying to make a connection. Is this the same type of thing that can get, they can be phosphorylated and then it can be used to direct where something ends up in the cell?
So there are many modifications that can help determine where a protein is going to end up. Some of these are actually built into the sequence of the proteins themselves. Some of them are modifications that are happening along the way, as we see here. So there's a variety of mechanisms for determining where a protein ultimately will end up. Okay. Okay. Yes?
All I was thinking about sort of like the antigens and how they can signal the like photoreceptors from blood types. Yeah, essentially.
Okay, so the question was what? I'm not sure I understand what it was.
If you like talk about that and how some of the glycoproteins are involved.
Oh, okay. So glycoproteins are, I think I see what you're saying. So when we look at glycoproteins, I didn't say this in class. One of these things you get in a review session, you remember you didn't say in class. But again, I'll tell you here, it's kind of interesting. If we look at a cell membrane, all right, and we look at the orientation of the glycoproteins that are there, every single one is oriented so that the carbohydrate part is sticking out. They're all sticking out. None of them are sticking in. And so that tells us something that partly that oligosaccharide part is important for the outer surface of the cell. And one of the places where that outer surface of the cell becomes up and becomes important is in identity, which is, I think, what you were asking and what you were asking also. All right? That identity helps an organism to tell, is this part of me or is this not part of me? Okay? So we may see some specific modifications that are happening on your cell or certain cell glycoproteins that you have, but not happening on somebody else's. And the issue with that is that your immune system actually evolves. When we talk about evolution, your immune system evolves during your lifetime. And during the evolution of your immune system, antibodies that attack your own proteins, structures in your own proteins and your own glycoproteins, those antibodies are selected against and they never appear again. All right? And we'll talk about this a little bit next term. Those that would provide some binding for things that aren't you, your body makes and they are continued to be produced. Okay? Now, why do I say that? Well, if you, for example, have a malfunction of the immune system and that selection process doesn't occur properly, you will develop what's known as an autoimmune disorder. Okay? And when that's happening, what the body is making antibodies that are attacking structures that are native to that body itself. So there are several autoimmune disorders that are known. Lupus is one of the more common ones. Psoriasis is another one, and there are others as well. Okay? Those things are antibodies attacking cellular structures that otherwise it wouldn't. Now, when we get the reason I bring that up is we talk about blood groups. The blood types that we have arise from specific oligosaccharides that are on the surface of our blood cells. And we have evolved our immune system so that we're not attacking that antibody, that antigen, that is the oligosaccharide that we have there. We're not attacking that, because if we did, we wouldn't be around for very long. We would be attacking all of our blood cells and we would die. Okay? So the fact that we're here proves that we've evolved that process of not attacking our own blood cells. Well, you put in a different blood type that has an antigen that your immune system recognizes, all right, you're going to get a very adverse reaction. And again, that could kill you as well, because now you've got a different blood type in there. The immune system is attacking it and plugging up your system big time. Okay? So you might say, well, what's a universal donor? What's a universal recipient, and so forth? So a universal donor is basically somebody who has what I would describe as a fairly naked oligosaccharide. It doesn't have parts of it that stimulate the immune system. Okay? So I'm an O-. O- is a universal donor, because my surface of my glycoproteins in my blood cells doesn't have anything on it that really stimulates an immune response, and my blood could go into almost anybody's blood in here without being attacked. All right? On the other hand, A and B instead have a real ability to stimulate the immune system. So if I took blood from a person with A and put it into my body, I would develop an immune system reaction against it. Make sense?
You talked about organ transplant.
Yep.
I'm not sure if you talked about anti-rejection drugs and how they work.
Yeah. There's a good reason why. So he said I talked about organ transplants and how you can get rejection, but I didn't talk about anti-rejection drugs, and I said there's a good reason why. One of the strategies of anti-rejection drugs is simply to reduce the efficiency of the immune system. So at a high level, the immune system is going to make antibodies against anything that it can and will. Reducing the effectiveness of the immune system is one strategy for reducing the likelihood of rejection. And that's one strategy for anti-rejection drugs. That's a bigger topic than I can go into here, though.
Imagine those people when they get infected more aggressively.
Yeah, it's a good question. Wouldn't people on anti-rejection drugs either get infected more readily or perhaps be more prone to develop cancer? I would argue that they would, but I don't know the clinical data on that. Yes?
So we're talking about oligosaccharides, and I know that has a bearing on like the fluviar or the
Okay, yep.
Yep.
So oligosaccharides relative to the flu virus. Okay? So let's go back to that figure that I showed in class talking about the flu virus. And there we go, right there. Here's the flu virus. And the flu virus is a complex creature that you can see. It has 1, 2, 3, 4, 5, 6, 7, 8 nucleic acids in this one. That's not a given. Flu viruses mix and match their genetic material from one to the other very readily. And not only do they swap individual pieces of DNA, they also swap within pieces of each other. So flu virus is a great example of a virus that's constantly evolving. It's why you get a flu shot every year, because the flu virus that's going to be here next year is not the same as the flu virus that was here this year. And it largely is working by attacking blood cells. Okay? So the flu virus attacks blood cells, and it does it by, first of all, grabbing onto specific oligosaccharide receptors on the surface of the blood cell with a protein that the flu virus has called hemagglutinin. And again, name tells us what it does. Heme referring to blood, agglutinin referring to agglutinate, stick to, right? So the hemagglutinin grabs a hold of this specific oligosaccharide on the surface of the blood cell and injects its viral RNA, flu viruses have RNA, injects its viral RNA into the cell. The viral RNA goes and starts replicating, starts making its own proteins, and some of those proteins will help to make up a new capsid, that is a new protective coat for the newly made viral RNAs that are there. Okay? In order for that flu virus to exit the cell, it's essential that this enzyme, neuraminidase, actually cleave on the inside a structure, I shouldn't say on the inside, but cleaves a structure in the membrane that has this an oligosaccharide in it. That neuraminidase action is necessary for the exit of the virus. So if you can stop the neuraminidase from acting, you can reduce the effectiveness of the flu virus to spread because it doesn't get out of the cell. All right? So neuraminidase inhibitors are used to fight the flu. And one of those I talked about in class is called Tamiflu. Tamiflu is a neuraminidase inhibitor. Yes, Eric. The nucleic acid for the flu virus is RNA. Uh-huh. Would an endonuclease work with a flu virus? Remember, we're talking about a human cell, and we don't have endonucleases, number one. But if we tried to cut that with an endonuclease, it would not cut it. That's correct. There are enzymes that will cut RNAs, though. All right. But they're a little different in strategy. Everybody tired and ready to go home? I know I am.
Is Tamiflu over-the-counter or prescription?
Tamiflu is prescription as far as I know. Omar, yeah.
Tell us what ring structures we're expected to memorize for the midterm.
So what, you're talking about what sugars I expected you to memorize? Well, so any of the sugars that I mentioned that you need to know the structure of, you need to know the ring and the straight chain. So that would be glucose, fructose, galactose, sucrose, and ribose. Mm-hmm. And fructose, yeah. And deoxyribose by extension, because that's, you know, the structure of ribose you've got deoxyribose, yeah.
Did you do a quick once-over of blood clotting?
Of what?
Blood clotting.
Blood clotting, okay. So blood clotting is, I think, a pretty amazingly orchestrated process. Blood clotting happens as a result of action of a cascade. And so it's the first example we've talked about with respect to amplifying a signal by starting with a very simple sort of thing, and at each step we see a great increase in the amount of each thing that's being made. So as each enzyme is being made further and further along, we're making this signal happen very, very quickly and very, very large. This quick action and this largely amplified signal is necessary to convert a monomer of fibrin into a polymer, or starting as fibrinogen, of course, but converting fibrinogen into a polymer of fibrin, right? And that has to happen before we bleed to death. And this guy right here, any of these guys on here, are so tiny to be invisible to the naked eye by a long ways. When you finally see a clot, we're talking about thousands or millions of these things being put together. So making this process happen very rapidly and very effectively and efficiently is critical for us to stay alive. I said we needed to focus down here because that process gets a little long in the tooth, all right? And the important things are happening at the very end. At the very end, we see the conversion of prothrombin to thrombin happening as a result of action of a protease. A protease is cleaving thrombin, doesn't matter which one, but it's cleaving prothrombin to activate it and make thrombin. Prothrombin being the zymogen form, thrombin being the active form. This is happening at the site of the wound. And it's important that prothrombin be at the site of the wound. And I talked about how prothrombin is held at the site of the wound by virtue of the fact that it gets its glutamate side chains carboxylated. An extra carboxyl gets put onto them, and that's a reaction that requires vitamin K. So prothrombin binds to calcium after it's been carboxylated. It stays near the site of the wound, and at the site of the wound, this process is going to happen to help it become thrombin. Thrombin, in turn, is a protease that cleaves fibrinogen to make fibrin. Fibrin, of course, is the monomeric unit of what will ultimately become the clot. Fibrin, in fact, will make a clot like, well, let's go back one. I don't want that quite there. This is the structure of fibrinogen, and as I noted, we have this big B unit that is basically blocking the ends of these things from doing anything. And down here we have this big A unit that's blocking these ends from doing anything. A is part of the alpha chain, B is part of the beta chain. Okay? The alpha chain when it has had its A units clipped off, the alpha chain can fit into these holes in the gamma units, and the betas can fit into their own, into the holes in the beta units. All right? So when that happens, we start to see polymers forming, and the polymers look like what's shown in the next slide, where we see in this case the alphas and the gammas are all joining. And so we see a sort of a network of this polymer that's starting to form. This figure doesn't show the betas fitting into each other's holes like here. And as I noted in class, this actually can happen in 3 dimensions. It actually can come out at you, so that the clot is a 3-dimensional structure held together by these interactions. The first part of the clot that forms is what's known as the soft clot. And the soft clot is just simply held together by these associations. These are not covalent bonds. These are simply interactions between these guys, hydrogen bonds, for example. Okay? Maybe ionic attractions. To harden the clot, there's an enzyme called transglutaminase that makes a covalent bond between glutamines and lysines. okay, between glutamines and lysines. And that will in turn cause these chains to be held together very tightly, as we've seen covalent bonds will do. So that's what happens to the clot, your clot, when it is getting hard and solid and crusty. Yes?
Question. You talk about how the transglutaminase is trapped in the local area of the clot, and I wasn't very clear on that.
So I'm not sure I understand the question. His question is, I said that transglutaminase is in the area of the clot, and that's right, it is. So
How was that related to it being near the clot instead of being all over your body and doing stuff that it shouldn't be doing?
okay, so his question is, how is transglutaminase held in the area of the clot? I haven't talked about that. There are other kinds of carboxylations that happen besides those for prothrombin. And I can't recall off the top of my head, but I think transglutaminase may be one of those things that's also carboxylated so that it binds calcium and stays at that site. But don't quote me on that. I think that's the case. Okay?
Yeah. Did you talk about hemophilia? Are we going to be responsible for that?
Well, I guess what I would say, go back and look at the videos. But I haven't specifically talked about hemophilia. I mentioned it in class, and I mentioned that if you imagine deficiencies in enzymes in that cascade, then if we can't go through that cascade, we're going to have troubles with respect to being able to form clots. But I'd say refer to the video otherwise. You had a question back here?
Yeah, back to the blood. What is the name of the enzyme that requires vitamin K and it causes
Yeah. So her question is, what's the name of the enzyme that uses vitamin K and puts those carboxyl groups on the prothrombin? I didn't give you that name.
I thought it was transglutaminase.
No, transglutaminase is the enzyme that's making cross-links between this and this. That's not vitamin K. That's not what vitamin K is doing. Vitamin K is stimulating the addition of carboxyl groups to the side chains of glutamates. That's a different thing. Omar?
So the side chains of prothrombin are carboxylated, and then prothrombin binds calcium.
So after the glutamate side chains of prothrombin have been carboxylated, then it can bind calcium. That's correct. So its binding of calcium helps it to stay localized to the site of the wound because damaged tissue releases calcium.
Okay, and the prothrombin there, and it has to wait for the cascade to get there, so prothrombin depletes and becomes thrombin. Okay.
Yep.
Is the reason that calcium is released from cells because the damaged cells have just ripped open their endoplasmic?
Exactly.
It's not
Exactly.
like death throw of the cell says, I'm dying, throw out calcium.
Yeah, it's a good question. So part of the reason that there's calcium present is exactly what you said. You ripped open those cells, and the things that had nicely sequestered calcium no longer have that. Yep. There was a question. Oh, so I guess I got to answer her question. Okay. Other questions? Yes? How does the clot break down? So the clot breaks down as a result of action of another protease called plasmin. Plasmin will actually attack the clot and start breaking it down. So it's something that you also want to regulate because if you don't regulate plasmin, you're going to be breaking down clots where you don't want to be breaking down clots. All right. So again, and I'm giving you a very simple picture. I don't claim to. I'm glossing over some things about blood clotting. I mean, there are entire courses taught on blood clotting, and we're talking about it in half a lecture that we can't go into here. Okay. So plasmin is a protease. It attacks the clot. It will dissolve the clot. And controlling plasmin is important. So you don't want to have it indiscriminately active, as I noted. And the control of plasmin is this protein that I described as TPA, tissue plasminogen activator. And what it does is it converts plasminogen into plasmin. So plasmin is made as an inactive zymogen, all right, as plasminogen. TPA is what causes it to become active. Make sense?
And through some method that you haven't told us, TPA is released.
Yeah, TPA. Actually, TPA is another one of these that is, I believe, also carboxylated, just like the prothrombin is. Okay, But the timing of that, I think, is critical, and I can't tell you what that is. Yes? Yes?
Is that just like alpha, alpha, beta, beta, and alpha, beta? Or do we actually know lactose, maltose? I know we need those sucrose.
So I guess the answer is you should be able to at least tell me the linkages of the sugars that are there, yes.
Okay, but we don't need to memorize lactose and maltose. Let's memorize the structures.
You're not memorizing the structures of those, no. No. Yeah. Unless you want to. Okay, folks, let's call it an evening.